Hi, morning everyone. I'm Boon from the Department of Molecular Medicine. So thank you for joining us this morning for breakfast at UMHealth. It's my pleasure to introduce our first speaker for today's sessions, Professor Dr. Azlina Aziz from the Department of Molecular Medicine. Prof. Azlina graduated with a Bachelor of Science in Biochemistry from University of Malaya and obtained her Master in Human Nutrition and PhD in Nutritional Biochemistry from the University of Glasgow. With over 20 years of experience in academia, Prof. Azlina has been actively involved in both teaching and research. She teaches biochemistry to medical and dental students at both the undergraduate and postgraduate levels. Prof. Azlina's research interest is in the field of nutritional biochemistry, redox biology, and natural products discovery. Her research group is interested in identifications and isolations of bioactive compounds from plant sources and establishing their mode of actions as antioxidants and how they protect cellular components against oxidative damage. Her research group is investigating how some of the antioxidant-rich plant extract may affect signaling pathway involved in disease development. Together with her collaborators, she's also studying the effect of redox imbalance in patients with several disease conditions, including thyroid disorder, cancer, and heart disease. Their group employs current technologies in their research, including genomics, proteomics, and metabolomics. And today, she will share her research journey on Tamarind in a talk entitled Unlocking the Biological Secret of Tamarind, a Decade of Research and Discovery. So just a housekeeping. If you have any questions during the sessions, please feel free to type them in the chat box and they will be addressed during the Q&A session later. And without further ado, the floor is yours, Prof.
Thank you, Boon. Let me quickly share my slide. Assalamualaikum and good morning, everyone. So in today's seminar, I will be sharing on the research that our group has done looking at the biological effects of the tamarind plant. So why we chose tamarind? Well, it is because when we started our work on natural products, we screened more than 20 local medicinal plants and we found that tamarind has strong potential based on their antioxidant activities and phytochemical content. Also at that time, research on tamarind was quite active and many researchers were working on providing scientific evidence for their medicinal claims. So let me first provide a quick introduction to the tamarind plant. So I think many of us are familiar with tamarind as we use the fruit pulp a lot in our cooking as flavoring due to its sour taste. But beyond that, it also has other beneficial functions. Let's first look at their botanical profile. So in terms of their botanical profiles, tamarind, or scientifically known as Tamarindus indica, is a fruit tree member of the family Fabaceae. It is a long-leaf and evergreen tree and can grow up to 30 meters tall, so it is a huge tree. So tamarind is native to the Eastern African region, but currently it is distributed in the tropical regions around the world, Malaysia including. So the fruit is the most valuable part of the tree due to its culinary, medicinal, and industrial applications. So these are some of their general uses. So the fruit pulp is used as ingredients in various culinary preparations, such as in juices, jams, and pickles, and as flavor enhancer. Its seeds can be roasted and consumed as snacks. The seeds are also used as cattle feed, while the kernel powder is used in the textile industry. Nutritionally, tamarind contains high concentrations of carbohydrates, dietary fiber, and proteins. It is rich in essential minerals such as potassium, phosphorus, and magnesium, and it is also a rich source of vitamins, particularly ascorbic acid and the B-complex vitamins. Tamarind contains phytochemicals with health properties such as polysaccharides and polyphenols. So this slide shows some of the traditional medicinal uses of tamarind as well as scientific studies for their health properties. So traditionally, different parts of tamarind are used in digestive health, for fever and infection, for treating malaria, particularly in regions of the world where there is a high case of malaria, for treating respiratory problems as well as for its wound healing properties. So over the years, various studies have been conducted to scientifically prove the medicinal claims of tamarind. And indeed, numerous studies have shown various parts to have antimicrobial, antioxidant, anti-inflammatory, cardioprotective, anti-hyperlipidemic, anti-cancer, as well as hepatoprotective properties. Because of the great interest on the health benefits of tamarind and the limited scientific knowledge on their biological properties, our research group decided to work further on this plant. So this slide outlines the overall workflow of our tamarind research, and which also reflects the typical pipeline for investigating the biological properties of a natural product. So we began first by optimizing the extraction process and characterizing the bioactive compounds present in different parts of the plant. Next, we evaluated their biological activities using both in vitro and in vivo approaches. For the in vitro approaches, we use both cellular and non-cellular methods, and for the in vivo work, we use animal models of specific diseases. And to move beyond conventional biochemical analysis, we also incorporated omics technologies to elucidate the molecular mechanisms underlying the observed health benefits. So the first phase of our research focused on polyphenols, which is one of the major classes of plant phytochemicals. Although tamarind contains various bioactive compounds such as carotenoids and saponins, we focus on polyphenols because of their well-established antioxidant properties and broad range of biological activities. So let me provide a brief introduction on what polyphenols are and why they are special. So polyphenols are a large and diverse group of naturally occurring secondary plant metabolites. They are particularly rich in many fruit and vegetables such as berries, in green tea, herbs and spices, as well as in nuts and seeds. So polyphenols can be divided into 5 major classes, which are the phenolic acids, flavonoids, stilbenes, tannins, and lignans. And within these classes, examples of more common polyphenols include quercetin, which are found mainly in onions and apples, curcumin, which are found in turmeric, and catechins, which are high in tea and cocoa. And research has shown that polyphenols have several key biological properties, including antioxidants, anti-inflammation, antimicrobial, and are able to modulate the immune system, cellular signaling, and gut microbiota. In addition, polyphenols have also been shown to be protective against diseases such as heart disease, cancer, and diabetes, likely due to these biological effects. So moving on to the first phase of our work. So one of the first things we did when we started the work on tamarind was to look at which part of the plant has the highest polyphenolic content and antioxidant activities, and which solvent is the most optimal for extracting the plants to give the highest activities. So the aerial parts that we work on was the leaves, the seeds, the veins, as well as the skins. And we use solvents of varying polarity to extract the tamarind plant. We then measured antioxidant activities by estimating their ferric reducing and radical scavenging abilities. We found that among the various parts of tamarind, the leaves contain the highest polyphenolic content and antioxidant activities, followed by the seeds. And we found that methanol was the optimal solvent for extraction of the bioactive polyphenols. Thus, our subsequent research uses these extraction methods for preparation of our extract. We next perform chromatographic analysis to identify the major polyphenols that are present in different parts of the tamarind plant, and we work on the leaf, the seeds, and the fruit pulp. Our analysis revealed a rich diversity of polyphenolic compounds, including catechins and procyanidins, which are well known for their potent antioxidant and other biological activities. So having characterized the phytochemical composition, we then investigated the biological properties of each plant part. So in the following slides, I will present our findings according to the plant part, starting with the fruit pulp, followed by the leaves, and finally the seeds. So let's first look at the biological activities of the tamarind fruits. So we first investigated the fruit pulp using a HepG2 liver cell model to examine its effects on gene and protein expression through transcriptomic and proteomic analysis. These omics approaches provide a comprehensive overview of the molecular pathways regulated by tamarind, rather than focusing on a few selected biomarkers. So on the left-hand side is the transcriptomic results. So transcriptomic analysis identified over 1,000 differentially expressed genes following treatment with tamarind fruit pulp. So pathway analysis highlighted two major biological processes of interest, which are oxidative stress response, and lipid metabolism. And among the upregulated genes were metallothioneins and glutathione S transferase, which are involved in cellular defense against oxidative stress. Additionally, we also observe upregulation of several lipid metabolism genes, including ApoA4, ApoA5, ABCG5, and downregulation of MTTP. So these genes regulate different processes of cholesterol and lipid metabolism, including reverse cholesterol transport. They also regulate cholesterol excretion as well as triglyceride metabolism. So these findings suggest that tamarind fruit pulp may possess hypolipidemic properties, prompting us to investigate this further. So we next perform proteomic analysis, which is the results shown on the right, which identified 4 differentially expressed proteins, including apolipoprotein A1, which is the principal protein component of HDL. And bioinformatic analysis indicate that these proteins were primarily associated with lipid metabolism and inflammatory pathways, particularly through this LXR, RXR signaling. So the LXR, RXR pathway is a master regulator of cholesterol, fatty acid, and glucose homeostasis, thus providing a plausible molecular mechanism underlying the lipid-lowering effects of tamarind fruit. So together, the transcriptomic and proteomic findings provided the first molecular evidence that tamarind fruit pulp modulates lipid metabolism and oxidative stress pathways, leading us to examine its hypolipidemic effects in vivo. So since our cell culture studies suggested that tamarind fruit pulp may protect against oxidative stress and possess hypolipidemic properties, so to validate these findings, we conducted an in vivo study using a hamster model of diet-induced hyperlipidemia. Also at this time, there was another study from Brazil who also reported the lipid-lowering effects of tamarind fruit using a hamster model, but they did not delve into the underlying molecular mechanisms. So we therefore aim not only to confirm the hypolipidemic effects in vivo, but also to elucidate the molecular mechanisms involved. The hamsters were fed a high cholesterol diet to induce hypolipidemia and then treated with tamarind fruit pulp for 10 weeks. And at the end of the study, blood and liver samples were collected for analysis of lipid profiles, antioxidant status, as well as transcriptomics and proteomic changes. So this slide summarizes the effects of tamarind fruit pulp on oxidative stress and lipid profiles. So as shown in the table on the left, tamarind treatment could restore antioxidant enzyme activities to near normal levels and reduced hepatic oxidative stress, indicating improved antioxidant defense. So there were 3 antioxidant enzymes that we measured, which were the catalase, superoxide dismutase, and glutathione peroxidase. And in terms of lipid profiles, which is shown on the diagram on the right, tamarind significantly reduced triglycerides, total cholesterol, LDL cholesterol, while HDL cholesterol remain unchanged. And these findings demonstrate that tamarind fruit pulp exerts both antioxidant and hypolipidemic effects, primarily by enhancing endogenous antioxidant defenses and reducing lipid peroxidation, resulting in improved lipid homeostasis. So to better understand how these physiological effects were achieved, we next examine the molecular pathways that is regulated in the hamster model. So this slide shows the hepatic expression of key genes involved in lipid and cholesterol metabolism in the tamarind-treated hamsters. Several genes were selected based on our earlier cell culture findings to validate whether the same molecular changes occurred in vivo. Importantly, ABCG5 and MTTP showed the same pattern of regulation observed in HepG2 cells, providing strong evidence that tamarind consistently modulates lipid metabolism across both experimental models. Additionally, we also observed upregulation of ApoA1 and LDL receptor, together with downregulation of HMG-CoA reductase, the latter being the rate-limiting enzyme in cholesterol synthesis. ApoA1 is found in HDL and plays a role in reverse cholesterol transport, while LDL receptor helps to remove cholesterol from the blood circulation. Their high levels are generally associated with lower risk of heart disease. So collectively, these changes indicate that tamarind fruit pulp lowers blood lipids through multiple complementary mechanisms, including enhancing reverse cholesterol transport, promoting hepatic cholesterol uptake and excretion, reducing cholesterol synthesis, and limiting triglyceride accumulation. So these molecular findings provide mechanistic support for the improvements in lipid profile that we observed in the animal study, linking gene regulation with the physiological hypolipidemic effects of tamarind fruit pulp. So in addition to gene expression, we also perform proteomic analysis to identify proteins altered by the tamarind fruit pulp treatment. Based on our analysis, we identified 4 proteins with increased abundance in the treated hyperlipidemic hamsters. And these are ApoA1, antithrombin 3 or AT3, transferrin, and vitamin D binding proteins. Although these proteins have diverse biological functions, they are all associated either directly or indirectly with cardiovascular protection, including in lipid transport, antioxidant defense, anticoagulation, and anti-inflammatory processes. Notably, the increase in ApoA1 protein was consistent with our earlier finding of ApoA1 gene upregulation, providing complementary evidence from both transcriptomic and proteomic analysis. And we also performed bioinformatic analysis, which showed that the altered proteins were predominantly involved in lipid metabolism, molecular transport, and small molecule biochemistry. And importantly, LXR-RSR signaling emerged as one of the top canonical pathways, the same pathway identified in our earlier HepG2 proteomic study. So the consistent identification of this pathway in both cellular and animal models strengthens the evidence that LXR-RSR signaling is a potential key molecular mechanisms underlying the hypolipidemic effects of tamarind fruit pulp. So in summary, our in vitro, in vivo, and omic studies consistently demonstrate that tamarind fruit pulp possess significant hypolipidemic activity. In addition, our study also provides mechanistic insight into its mode of action. We propose that its polyphenolic-rich composition, including catechin and epicatechin, modulates the expression of key genes and proteins involved in cholesterol and triglyceride metabolism. These molecular changes promote reverse cholesterol transport, reduce cholesterol synthesis, and enhance triglyceride metabolism, resulting in lower circulating cholesterol and triglyceride levels. Our findings also further suggest that these effects are mediated, at least in part, through activation of the LXR-RXR signaling pathway. In parallel, antioxidant properties of the fruit polyphenols help protect cells against oxidative stress, providing an additional mechanism for their beneficial effects. So together, these findings provide molecular evidence that tamarind fruit pulp exerts both hypolipidemic and antioxidant effects through coordinated regulation of multiple metabolic pathways. So we next investigated the biological activities of the tamarind leaves. So although this work was less extensive than our fruit studies, our preliminary analysis indicated that the leaves possess strong antioxidant activity. So to validate this, we used a HepG2 cell model of hydrogen peroxide-induced oxidative stress. So pre-treatment of the cells with the leaf extract significantly protected the cells against oxidative damage. Specifically, the extract reduced intracellular reactive oxygen species and lipid peroxidation, while restoring the activities of the antioxidant enzymes, mainly superoxide dismutase, catalase, and glutathione peroxidase. In fact, in some cases, to levels even higher than the untreated controls. This finding suggests that the polyphenol-rich leaf extract enhances endogenous antioxidant defenses, thereby reducing oxidative stress and protecting cells from oxidative injury. And these results identify tamarind leaves as a potent source of natural antioxidants and support their potential role in protecting against oxidative stress-related diseases. To gain further mechanistic insight, we also examined the gene expression profile of HepG2 cells treated with the tamarind leaf extract. Transcriptomic analysis identified several upregulated antioxidant-related genes, including glutathione transferase, aldehyde dehydrogenase, and alcohol dehydrogenase, which are involved in cellular defense against oxidative stress. Interestingly, these genes differed from those regulated by the fruit pulp, suggesting that the leaves and fruits may exert their protective effects through distinct molecular mechanisms. In addition, bioinformatic analysis identified the top canonical pathways as the coagulation system, cholesterol biosynthesis, and intrinsic prothrombin activation pathway. Although we did not pursue the leaf study any further, identification of coagulation-related pathways was interesting, as it suggests a previously underexplored role for tamarind leaves. Overall, these findings suggest that tamarind leaves possess potent antioxidant activity and may influence coagulation pathways, providing new directions for future research. So we next move on to the biological activities of the tamarind seeds. So we next evaluated the antioxidant effects of tamarind seeds using the same HepG2 cell model of oxidative stress. Similar to the leaves, the seed extract protected cells against oxidative damage, and this protection was associated again with increased activities of the antioxidant enzyme, superoxide dismutase, catalase, and glutathione peroxidase, resulting in lower reactive oxygen species and reduced lipid peroxidation. We propose that these protective effects are largely attributable to the high polyphenol content of the tamarind seeds. These findings identify tamarind seed as another potential rich source of natural antioxidants and provided the basis for our subsequent studies on their anti-diabetic potential. So tamarind seeds have been widely reported to possess anti-diabetic and hypoglycemic activities. So to identify the research gaps and understand the proposed mechanisms, we conducted a systematic review of the available evidence. And our review showed that tamarind exerts anti-diabetic effects through multiple mechanisms, including reducing oxidative stress and inflammation, regulating glucose and insulin metabolism, improving insulin signaling, as well as modulating glucose digestion and absorption. And among the different plant parts, tamarind seeds demonstrated the strongest anti-diabetic effects. However, the review also revealed a lack of molecular evidence explaining these mechanisms. Therefore, this knowledge gap formed the basis of our subsequent study, in which we used a type 2 diabetic rat model to elucidate the molecular mechanisms underlying the anti-diabetic effects of tamarind seeds. So to investigate the anti-diabetic mechanisms of tamarind seeds, we established a type 2 diabetic rat model using a high-fat diet and low-dose streptozotocin. Diabetic rats were treated with low and high-dose tamarind seed extract for 28 days, after which blood and tissues were collected for biochemical, transcriptomic, and metabolomic analysis. We found that tamarind seed, particularly at the lower dose, significantly improved glycemic control, suggesting a dose-dependent effect. And gene expression analysis indicated involvement of insulin signaling pathways, while metabolomic profiling revealed alterations in glutathione metabolism and arachidonic acid metabolism, suggesting antioxidant and lipid signaling roles. So these findings provide preliminary molecular evidence that tamarind seed improves glucose homeostasis by modulating insulin signaling and key metabolic pathways in type 2 diabetes. And overall, this result extend our earlier findings and demonstrate that tamarind seeds not only possess antioxidant activity, but also show considerable promise as a source of anti-diabetic bioactive compounds. So in summary, our research has advanced the understanding of the molecular mechanisms underlying the biological activities of tamarind by integrating omics technologies with conventional biochemical approaches. So we demonstrate that the tamarind fruit pulp possess hypolipidemic activity, likely through regulation of cholesterol metabolism and triglyceride homeostasis. We showed that the tamarind leaves exhibited potent antioxidant activity and showed potential to modulate the coagulation pathways. And we showed that the tamarind seeds displayed hypoglycemic effects with molecular evidence suggesting involvement of the insulin signaling pathway. The diverse chemical constituents, including polyphenols, likely contribute to tamarind's observed biological activities through synergistic and multi-targeted mechanism. Together, these studies demonstrate that tamarind is more than a traditional medicinal plant. It is a promising source of bioactive compounds with scientifically supported mechanisms that warrant further translational and clinical investigation. So where do we move from here? So the ultimate goal is to translate these promising findings into well-designed human clinical trials. However, several key knowledge gaps must first be addressed. First, there is limited standardization. of tamarind extract due to differences in plant parts and extraction methods. So developing a standardized extract with robust quality control is essential. Secondly, many proposed mechanisms remain supported mainly by biochemical evidence. So applying omics technologies will provide a comprehensive systems level understanding of tamarind's molecular mechanisms. Third, although existing studies suggest a favorable safety profile, more data are needed on long-term toxicity, safety, and potential herb-drug interactions before widespread clinical use. And finally, research has focused predominantly on the fruit, while the seeds, the leaves, and the bark remain relatively underexplored, despite their rich phytochemical composition and promising bioactivities. Therefore, future research should focus on standardization, mechanistic studies using omics technologies, comprehensive safety evaluation, greater exploration of underutilized plant parts, and rigorous human clinical validation to fully realize the therapeutic potential of tamarind. So before I end my talk, I would like to take this opportunity to thank all the people that has contributed towards making this research possible. So these are my research collaborators, Prof. On, Prof. Sarni, Prof. Putri, Dr. Jamie, and Prof. Azhar, who are experts in their own research areas, as well as 4 of our PhD students, Nani, Choi Yin, Ursula, and Intan, who work on different parts of the tamarind plant. With that, I thank you. Assalamualaikum.
Okay. Thank you, Prof. Azlina, for the insightful presentations and also for sharing your research journeys. It's like a very good insight on tamarind, especially it's like before this, we only think that the fruit is beneficial, but now we know that actually the leaf and the seeds also have the very good medicinal properties. So now we will move on to the Q&A sessions, and I think we have a question in the chat box. So I'll read out the question from Dr. Yu Guan Chan. How much tamarind do we need to benefit from this? Is eating the nasi lemak where the ikan bilis is cooked with tamarind enough?
Obviously, like what I mentioned earlier in the limitations of study, mainly when the work that we do, because we are trying to determine the mechanism of action, obviously we tend to use a much higher dose so that we can actually see the effects that we want. So obviously, if we want to translate this into human consumption, of course we cannot directly translate what we found in our lab work into human consumption. So that's where standardization of extract will be important. So if we say we are just consuming from the normal food that we eat, I would say that the phytochemical or the bioactive compound will actually be present in quite low amounts. But if we are looking at more of therapeutic effects, then that's where studies on pharmacological dose response studies would be important. Yeah, so I hope that answers the question.
Thank you, Prof. I think that's all the questions. But there's one anonymous attendee. It's like, how would AI change the way you approach the research?
Quite interesting question. Well, not in terms of how we conduct the work, but the way I see it is more of like how we can use AI to see compare like what other people have done, and then to probably determine what is the best approach or what is the current work that are being done, especially on natural products. So I don't know. Maybe that answers the question or not.
So I just have a general question for Prof. Because we now see that the leaf and the seed also is beneficial with the medicinal properties. So do you think people will accept to take in leaf or seed instead of the fruit tops?
Yeah, maybe not for consumption per se, but I think if you are to convert it into more of a supplements product where maybe turn it into a powder form or something, then it would be probably more edible, because generally people don't just consume seed as a normal diet, right? Because firstly, it doesn't have a nice taste, it's very bitter. I know some parts of the world, they do consume the leaves as like an ulam. Yeah, but I think the best way to make it more edible is to actually turn it into powder form.
So one last question, Prof. I just would like to know the sources of this fruit pulp of the tamarind as well as the leaf and the seed. Are they coming from the same source?
Yeah, actually
Because
we
I see
have,
that the time frame of the study are different.
yeah, actually they do come from a source in UPM. So they actually grow the plants, and that's where we get the source from.
Thank you, Prof. I think that's all from Prof. Azlina, and I'd like to thank Prof. Azlina again for the insightful research journey on the tamarind that she shared with us just now. And now we will move on with the next sections. I pass over to Dr. Nurul.
Thank you.